It's really great pleasure that we are doing a webinar, which is on an online platform from Diabetic Asia Study Group, along with IDF Southeast Asia region. We have with us our President-elect, IDF, Dr. Peter Schwarz. We have also with us our President of Diabetes Asia, Dr. Abdul Bashir. And we have with us IDF Southeast Asia Chair, Dr. Ajad Khan. We have many eminent faculties from different parts of the world and mainly from different parts of Asia. Dr. Bakkar, Dr. Sanjay from RSSDI, Dr. Vijay Vishwanathan from RSSDI. We have past president of DSG and past IDF Southeast Asia Chair, Dr. Anil Boraskar, and very eminent person from India, Dr. V. Mohan, who is also past president of DSG. We just now Shashank joined, who is past IDF Southeast Asia Chair. The idea of doing this webinar on glucagon, as we know that we have done a lot of activities when we were celebrating 100 years of insulin discovery. And then we realized that glucagon is equally important, and it was in 1923. So 100 years of glucagon discovery, we should do some activity. That was the complete idea. I first talked to Dr. V. Mohan and then Dr. Abdul Bashir. And we thought of that we will come out with a special issue also on this. And Dr. V. Mohan, Editor-in-Chief of Journal of Diabetology, which is a journal of our Diabetes Asia. Being I'm a currently Vice President of DSG and I'm also Chair-elect of IDF Southeast Asia. Then I called Dr. Ajad and they all agreed that yes, we should have a dedicated issue and there's a special issue on with and I just requested Dr. Mohan that we will get 10 article and we could get it. That was really, and thanks to everyone. And then Dr. Abdul Bashir had told me, yes, we should do a webinar also. And that's how this webinar is planned or rather formulated. We requested mainly Indian speakers and that too I could connect them easily. But we also tried to connect some of our colleagues from Nepal, like thanking Dina, Sharad from Qatar, and few more from other parts of the world. But Peter, Abdul, Ashar, and many of them who are part of our panel discussion. As glucagon, and it's very important clinically also. It's not like that it's clinically not important. That's the reason we designed the webinar in such a way that it should interest clinician. That was our primary idea. And when we started, it is a free registration, but maximum number of doctors, participants from India, who is more than 1,200 doctors who registered for this program. So I'm really very happy to share this number, these many doctors who have registered, even in this, virtual world, they have joined us. And then we have doctors from all over Asia and even from US and UK also joined. So that number also I shared before some time, it is more than 1,500 delegates from all over the world. And now it has increased to almost roughly 2,000. This year, simultaneously, we also have the birth anniversary of 100-year centenary year of Professor Vishwanathan, which we are celebrating. And we decided that last 3 months, October, November, December, when we have the World Diabetes Day month, which is World Diabetes Day Awareness Month, why not to celebrate different activities, not only for educational activities like webinar or some of the other activities which we are doing across the country. We are also doing with Diabetes India and RSSDI and many organizations locally also diabetes awareness campaign, which we have started doing. We are having our Indian Diabetes Risk Score. Long back, Dr. V. Mohan had published an article in JAPI. And on that basis, we have made an app and we have circulated this app to not only India, but our neighboring countries and our colleagues also. So they can also download and whatever number of patients or to laymen, to whomsoever they want there, what is the risk, they can calculate. So in this 2-3 months, we are going to do a lot of activities and all activities will be dedicated to Professor Vishwanathan, who is father of diabetology from India. So with these few words, I will request first Dr. Vijay Vishwanathan to talk few words for Dr. Vishwanathan, and then I'll be requesting Professor Peter, Professor Ajad, and Dr. Abdul Bashir, and then I will have few words from Shashank Joshi, V. Mohan, and Dr. Anil Boraskar. With these few words, requesting Dr. Vijay and requesting AV team to put the photo of Dr. Vishwanathan. Yes, Vijay.
Thank you, Dr. Banshi. And I can't name every dignitary who's on the screen, but I think we would like to thank you for remembering Professor Vishwanathan on his 100 years. And Professor Vishwanathan worked mostly on primary prevention of diabetes in the 1950s, and also the dietary habits of people with diabetes. And to talk about primary prevention in the 1950s is a real blessing in disguise, because he talked about hyperinsulinemia and about primary prevention at that time in the 1950s. And we owe a lot to him because he started the first diabetes clinic in India in 1948. So we would like to dedicate this program. Thank you, Dr. Banshi, once again for taking up this issue of glucagon, 100 years of glucagon. Thank you very much. Thank you.
Thank you, Vijay. Thank you for this. Dr. Peter, can I request you for a few words for this?
Yeah, dear Banshi and dear colleagues, I must say I'm totally fascinated by the opportunity to talk here in such a fast developed webinar about such an interesting topic. As Banshi Shabu already said, we all talk about insulin, and 100 years ago or 101 years ago, insulin was discovered, and since then we use it to treat diabetes, we use it to manage diabetes. And I must say personally, we actually have forgotten about the glucagon. And glucagon is such an important player for the diabetic paracrineopathy, for the development of diabetes, but also today as a player in the development of NASH and in terms of the effect of fasting. And that's why it's so important to discuss glucagon, or to revive the story of glucagon in diabetes management. So thank you very much to Banshi and all the colleagues. Thank you very much to DASG for installing this webinar. And may it be the starting point, or may it be a seed in a fertile soil to grow a better understanding of glucagon and its effect in the diabetes world, but also for metabolic syndrome and beyond. It's a great honor to be here today, and I wish a very successful and fruitful webinar.
Thank you. Dr. Akhtar video can we have? Thank you, Dr. Peter.
I'm Dr. Akhtar Hussain, the president of International Diabetes Federation, but most of all, I'm a friend of yours. I would like to, first of all, I'd like to congratulate all my friends here, especially Dr. Banshi Sabu and Professor Eka Azad Kansar for organizing this event. I would also like to thank the organizations like DASG, Diabetes Asian Study Group, IDF, Southeast Asian region, and also Diabetes India for organizing this very important event to mark the 100 years of glucagon. And we all know about the importance of the glucagon therapy. This was the year 2023, marks the 100 years since the publication of the first report of hyperglycemia factor in pancreatic extracts, which C.P. Campbell and John R. Murelin named glucagon from glucose agonist. Still, the work to fully understand the production and the biological effect of glucagon has proceeded at a slower pace compared to that of insulin. Glucagon, the principal hormone produced by pancreatic islets alpha cells, has a range of profound effects on metabolism, including but not limited to stimulation of hepatic glucose production. Accordingly, glucagon has been utilized as a first-line treatment for severe hypoglycemia since 1950s. The year 2023 marks 100 years since the publication of the first report of hyperglycemic factor in pancreatic extracts, which C.P. Campbell, John R. Murelli named glucagon. So the therapies that we are using based on glucagon is unlimited, and we all know that how important it is for saving the lives of the people with diabetes. We must not forget that this is also the year that when we also are celebrating or remembering with due respect the 100 years of Dr. Vans Vishwanath's life. He is considered the father and the most important personality in diabetology, endocrinology, not only in India, but in the whole subcontinent. IDF strongly supports this initiative. IDF strongly supports the activities of all these people, and I'm very proud to say that IDF Southeast Asian region seems to be quite active, and they are playing a very important role, particularly the time of World Diabetes Day. I know that you have many activities, and I wish you all success in your activities. Thanking you.
I will request Dr. Abdul Bashir if he is there. Yes, sir. Dr. Abdul Bashir is currently president of Diabetic Asia Study Group, and this we are organizing under the banner of Dr. Abdul Bashir, the DSG and IDF Southeast Asia Company. Dr. Bashir sir.
Hello everybody. It's my pleasure and honor to be the president of DASG at present, and I cannot express my gratitude in words, neither to Banshi and Bishu and others who have organized this webinar, nor to those my predecessors like Azad Saab, Mohan, Akhtar, and the unrestricted support we get from the platform of IDF, for which I'm grateful at present to Akhtar and now Peter Shavard. The important topic has already been highlighted and will be discussed in the next 2 days. I also remember late Prof. Shera and me visited Chennai, and we had visited Vijay's and Mohan's father's place, and the work he has done in the early 20s of 19th century, that was really remarkable and commendable. And as Akhtar has rightly said, that it's not only India who need to acknowledge his work, it's the whole subcontinent, and we, the whole region, acknowledge his work and his legacy he has left by leaving people like Mohan and Vijay, whom we take more as friends rather than as scientific legends, but in fact, they are legendaries in their respects. I, as president, also welcome all the fraternity who have joined, and my special thanks and gratitude to Banshi Sabu, who also wants to work all the time, all the while. I think he works 48 hours in a day, and people like me who are very old also keep dragging, maybe at least 12 to 24 minutes every day. So thank you all. Thank you, and looking forward for a very good seminar and webinar. Thank you.
Thank you, Dr. Abdul Bashir. Professor Azad Khan, sir, for your words of blessings.
Yeah, Bonsi, many thanks to you and your team. And also, I'm grateful they have invited me so that I can keep myself educated. It is really commendable that you have taken initiative to observe the 100th year of glucagon. I dare say the glucagon has been somewhat neglected to the diabetologists. The glucagon that is also very important, that needs to be reminded, that needs to be taken into account. But anyhow, I wish this conference webinar a great success. And I can see Mohan, as usual.
Thank you.
Thank you.
Thank you, sir. Now I request Professor Dr. Anil Boraskar, sir, who is a past president of DSG as well as past chair of IDF Southeast Asia, eminent diabetologist. Dr. Boraskar, sir.
Thank you, Dr. Bansi, and all my very dear friends from India and abroad. DSG is very dear to me. We started it together. I remember the first conference, second conference, third conference, and we had continuous encouragement and guidance from Professor Azad Khan. And we have people, I have to tell this, that Dr. Vishwanathan has got a very, I was special position for him in my life. Many years back, many years back, I had visited Chennai and came to him for his blessings. And he said, Diabetic Association of India needs a strong shoulder like you. And he gave me his blessings. And in 40 years, there is not a single day that I don't remember Professor Vishwanathan Mohan. You're so lucky that you have guidance from your great father, and we are all so grateful to him for shaping India's academic platform so well. And I am really grateful to Bansi for inviting me for this function. All my friends are here. Shashank, Makkar Sahib, Amit Gupta, Vishwanathan, Vijay. And I see their progress and they're doing so well, all of them. And we are very proud to have all this great faculty. I see Shweta from, I think, Nepal, if I'm not mistaken.
Dina. So she is Dina.
Zina, yeah. So nice to see now new faces, charming faces. And I'm very grateful to Bansi for inviting me in this. Thank you very much.
Sir, all your blessings, please. Now I request Dr. Makkar, who is currently president of RSSDA India, one of the biggest organization of diabetologist professionals from India and maybe third largest in the world after AD and PhD. Dr. Makkar for a few words.
Thank you, Bansi, and greetings from India to everyone from outside India. I think it is a great pleasure and honor to be at this platform, sharing, this platform with the who's who of the diabetes fraternity from the globe. And my, sincere thanks to Bansi, DSG, and our IDF Southeast Asia Regional Group for having me here. As Dr. Abdul Basit said, Bansi works 48 hours a day and nobody can match his energy. And he keeps pushing us to do more work. And I'm sure this webinar or this, whole event focused on glucagon is going to be one of the landmark events in today's era, because we always talk of so many things now, but still forget glucagon as one of the major players. So thanks, Bansi, and thanks everyone. Wish you all the success.
Thank you. Dr. V. Mohan, sir, for your blessing, and then we will start also. We have Shashank and Amit will finally thank. Yes, sir.
Thank you, Banshi. It's a great occasion when three different bodies, the International Diabetes Federation Southeast Asia, DASG, as well as Diabetes India, along with many other representatives. We have RSSDA represented by its president, and we have the president of the IDF itself here. So it's a great occasion, and I think I must give full marks to Dr. Banshi Sabu, because not only he, at very short notice, organized this meeting, and all of you said he works 48 hours. I think he works about 200 hours a day. Otherwise, I think even 48 hours is not enough to accomplish all this. In fact, just to give you a small digression, when he said that, he'll get 10 articles for JOD, a Journal of Diabetology on glucagon, I kind of laughed at it, because I said, where are you going to get? He told me this about 3-4 weeks ago. He said 10 articles in 3 weeks for a journal you're actually going to get them. And I didn't think it's possible actually as an editor, but he did it. And the articles and is right now under press, and many of you are authors in that. And it's amazing. I mean, what he does is amazing. Now, this tribute to my father, I think, is again his generosity, because for 3 whole months, all the activities of RSSDI and all the other World Diabetes Days, he has, kind of dedicated it to my late father, and both Dr. Vijay Viswanathan and myself, my brother, and myself, as well as Dr. Ramchandran, my brother-in-law, and the whole family is very, very grateful to you for the due respect that you have given to this great man, who was much ahead of his time in his thinking. And all the names you mentioned, Professor Samad Chera from Pakistan, and Professor Ibrahim from Bangladesh, and they're all dear friends, and they're all contemporaries, and therefore they've left a whole legacy which we are very lucky to follow. And as Peter Schwartz rightly mentioned, and he sent some beautiful slides and material, glucagon, actually the discovery of insulin and glucagon are almost the same, because it's at the same time when the contamination was also just thought to be a contaminant of insulin. They're trying to get rid of it. And then they realize a new hormone all by itself. And I'm going to talk about that. So I'm not going to take much more time, but all the luminaries here are all my close friends. Thank you all for participating in this meeting and for making this such a grand success. I look forward to the whole presentation, and thank you, Vanshi, for making me the opening batsman also, along with my good friend Dr. Shashank. Thank you.
Shashank, few words from you.
So, Bansi, first of all, congratulations to you for being super, superlative in many ways for organizing this meeting. We are all here for a common goal and a common cause. We are all here committed for people living with diabetes. And we are all here to celebrate this World Diabetes Month. So I first pay my regards to all the seniors here, particularly global people. I can see Peter, Professor Azad Khan, Professor Abdul Basit sir, and many others, Ashraf I can see. And then all my teachers here, Dr. Mohan, Dr. Anil Boraskar, and Dr. Vijay Vishwanathan, my good friend Amit, I can see Dina, and various others. So we have all here gathered after 103 years of insulin discovery. We are now focusing on glucagon. And glucagon is also 100 years old. So I'm very happy that we have rediscovered glucagon. And I'm grateful for Peter for reinvigorating some very insightful articles which he put out on the group. So thank you, Peter, so much for that. It's really insightful. Dr. Mohan has been very unique in many ways. But Bansi, you are truly unique. There's only one Bansi Sabu. And in the Indian culture, the Bansi stands for Lord Krishna. Okay, and this, Professor Azad Khan always knows more Sanskrit than Urdu sometimes. And, so Lord Krishna, does multitasking. He was the god of multitasking. And that is the name of Bansi Sabu. So he is the super tasker. And we are here to celebrate science and excellence. And we are here to learn. And whatever we learn, we are here to adapt. So I feel that this 2-day conclave will enrich all of us. I can also see a lot of other people like my friend Sanjay Agarwal. I can see Ashraf there after a long time. I can see Dr. Ashraf Fadawat. Sorry if I missed out somebody here in the crowd. So we are here to learn and adapt. And I think the glucagon story is just starting now. And it really threatens to make people lean and thin. I was elected Obesity Society president in India because they looked at my BMI. But this glucagon is one hormone or its peptides, single agent, dual agent, triple agent. They may actually threaten to make everybody on earth lean and thin. And this looks to be a reality. It can change the diabetes demography. It is already threatening food companies. So I am very grateful that Bansi is putting out this virtual educational exercise. And then he's collating and compiling it in the journal which Dr. Mohan is editing. And we have all the support from all over the world. So thank you so much and congratulations. And let's all get ready to learn some high impact science, which will make our ability to handle our patients better, because that is what we are committed for, and make lives of people with diabetes. Ultimately, that's what is our primary commitment. That is our God. I hope that they can have better lives, longer lives, and complication-free lives. Thank you so much.
Thank you. As we are running late, Dina, you want to say a few words? Actually, Dina is very active from Nepal Diabetic Association. Before Dina, nobody is very active from Nepal, and I always call Dina, to activate more and more physicians from Nepal. And we have few common friends and few industry friends also who are also working. So Dina, for a few words, and then I will request Amit for vote of thanks. And then we close because we are already late by 15 minutes. Dina.
So I would just say this is amazing, and I'm really glad that I'm part of this platform. I look forward to this learning, and I think everybody said whatever needs to be said. So thank you, Dr. Banji, for including me. And
thank you, Dina. Thank you. Amit, I request for thanks. We propose, and then we start with first talk also.
Thank you, Dr. Bansi sir, for giving me this opportunity. And I really feel humbled to be present among this constellation of stars who have shaped the future of diabetes, not only in India, but globally. I would like to thank Dr. Akhtar Hussain, Dr. Peter, Dr. V. Mohan, Dr. Professor Azad Khan, Dr. Abdul Basit, and all the faculties who have joined, Dr. Vijay Vishwanathan, Dr. Ashraf Fawad, Dr. Ashraf, and those faculties who are going to join tomorrow for the second day of the session. So I think we have interesting 2 days scientific sessions happening, and interesting story of the glucagon will be discovered, and we are going to learn a lot of many things. So with this, I would like to thank each one of you for your constant support and contribution. Thank you, sir. Thank you.
Thank you. So thank you, Amit. Amit is a big asset, sir, for India, and his support to me and to everyone is really commendable, and big round of applause for Amit also. With this, now we start with first talk. Today we have two talk with Dr. Mohan and Dr. Shashank will be talking, and then we will have a panel discussion. So can we have the slide for the chairperson? Neela? Yes. So Dr. Abdul Bashir and Dr. Ajad Khan, we all know them, and they are going to chair the session. And Dr. Bashir, should I request you to introduce Dr. V. Mohan? Everyone knows, but still there are. And just to, before I introduce Dr. V. Mohan and asking for that, just to tell, because there are more than 1,200 doctors from India who have joined already, and more than 250 from other parts of the Asia. Those who are not knowing much about the Diabetes Asia, it is a organization transnational with different countries as the part of it. An individual member can become the part of DSG. Please visit our website of DSG, and by paying $100, you can become the life member, and you can get the GOD also free. So those who are not the member, including faculty member, if they are not the member of DSG, please join DSG and make it bigger and bigger. With these words, now I request Dr. Abdul Bashir to introduce Dr. Mohan, and then we can start the talk. Over to Dr. Bashir, sir.
Abdul, I think we need not introduce. We can save some time. We can go ahead. We all know each other, and I think we can go ahead. You can say some other word, but need not have the introduction at all.
Right. I will only say that Mohan doesn't need no introduction, and
That's
once
enough.
you listen to him, you will never forget him anyway. So Mohan, over to you.
Thank you. Thank you. It's a great honor to have you and Professor Azad Khan, two of my closest friends in DASG, to chair the session. What I'll do is I'll share my slides first. And is it seen?
No, sir.
No. Okay. Okay. Let me minimize it. I'll put it again. Now?
Yes, it is. Yes.
Now it's seen. Okay.
Yes.
So, let me once again thank the two chairpersons, and special thanks to Banshi and Biswajit and Amit and all of you who are there, and again paying my respects to my father. So the topic I've been given is to talk about the history of glucagon with novel insights of its physiological function. I'll talk about the history and the physiological function. I'm not going to go into the therapeutic aspects of the drugs because there's a separate session on that tomorrow. And I don't want to steal the thunder by talking about that today. I have no conflict of interest to declare. Now, for those of you who have not read this paper, and I know that Peter also sent this article published in Lancet Diabetes very recently by our good friend and old friend Pierre Lefebvre and Sheen. And this is a fantastic article. If you've not read it, I think you should read it. It's very thrilling to read it actually. Now, this is a table which tells you that right from 1922-23, when Banting and Best discovered insulin, glucagon was also discovered, and they thought it's some kind of a contaminant which was there, and they were trying to get rid of it. And I think Professor Akhtar Hussain also mentioned a part about it. Now, it'll take me a long time to go through this thing, but you can see that, and parts of it I'm going to show anyway in my subsequent slides. But over the years, as has been repeatedly stressed, while insulin was given a lot of importance, this is a kind of stepmotherly treatment given to a stepbrotherly treatment given to glucagon. But it's only now that the newer agents have started coming that glucagon has again come into focus again, and we'll talk about that. Now, this was already mentioned by Dr. Akhtar Hussain, that is in 1923 that Merlin and colleagues found out this term of this hormone called as glucagon. which they coined this term because glucose and it's an agonist, therefore they called it as glucagon. And a decade later, after 1923, Berger and Brandt prepared a partly purified material, but 1948, and these are the big guys, and you'll see why in a few minutes when I talk about it. Sutherland and Dewey, characterized the origin, and they called it as a hyperglycemic glycogenolytic factor of the pancreas, which later was confirmed by Dewey in 1953. Even at that time, because insulin was being used and hypoglycemia was being developed, they started using this glucagon preparations in insulin reactions. And that's been one of the biggest uses of glucagon till today. To further go down, in 1955, Staub and colleagues purified it and crystallized glucagon, and the amino acid sequence was established by Bromer. And glucagon synthesis was achieved by Wunsch and Weins, and the structure of glucagon is established by Blundell. And they knew that it's a polypeptide made of 29 amino acids. Now, going on further into the cellular origin of glucagon, Sutherland and Dewey identified the alpha cells of the islets of pancreas in 1948. You can see it's already taken 25 years after the discovery for the cells to be identified. And then the localization of glucagon, the alpha cells, occurred in only 1962. The reason why it has taken so much time is because people are not worried about this. They thought it's some kind of a hormone, doesn't have much of effect. Why should you waste time on this? And even the industry was not interested in that. But then Lelio Orsi described the precise microanatomy for which, of course, he got the Nobel Prize. Now, Roger Unger was a good friend of mine. I've met him many, many years ago, and we kept in touch for some time. And he was later editing the Metabolism Journal. So he described the radioimmunoassay of glucagon in 1959, for which, of course, he should have got the Nobel Prize. But the problem was that Yalow and Burson had already described the radioimmunoassay of insulin a year later, and a great tribute to Roger Unger, because he acknowledged that he went to the laboratory of Burson and Yalow and learned the radioimmunoassay technique for them. So the Nobel Prize actually went to Burson and Yalow for describing the radioimmunoassay, which later, and so insulin came, and then later glucagon also came. The glucagon receptor, the GCGR, was later described, and they found that it was expressed mainly in the liver and the pancreas, of course, a little bit in the heart and adipose tissue and so on. Now, this GCGR encodes the glucagon receptor, which is a 62 kilodalton protein and a member of the class B G protein. And for discovering this G protein, Martin Rodbell actually got the Nobel Prize again. And these are the three Nobel Prize winners for glucagon research. Dr. Sutherland, Dr. Christian D. Dewey, and Martin Rodbell. In different years, they got the Nobel Prize. And so 3 Nobel Prizes have been given for glucagon. And I'm sure you know that for insulin, even more than that has been given. Now, the glucagon gene is located on chromosome 2. And this gene encodes the pre-pro hormone. And I'll show you this better in my next slide, because among them are the 2 glucagon-like peptides, the GLPs, which, of course, in recent years has become more famous because you have the GLP-1 receptor agonists. But then there are other members of the glucagon superfamily, like the glycentins. And if I show you this particular one, it's the pro-glucagon, which is the precursor of glucagon. And then you have this NEC1, the neuroendocrine converters enzyme 1, and the NEC2, which is a converter 2. And through the NEC2 processing from pro-glucagon, you get glucagon. And then, let me see if we can get a laser pointer. Yeah, you get glucagon. And from the NEC1 processing, you get the GLP-1 and the GLP-2, and you also get the glycentin. And from the glycentin, of course, you get the oxyntomodulin and the GRPP and so on. Now, let's confine our discussion today to glucagon, because tomorrow you'll be hearing more about the GLP-1 and the GLP-2 and so on in these subsequent talks. But I want to go down to some of the basics of glucagon, because this is what you should know. It's a peptide hormone secreted by the pancreatic alpha cells. It's a polypeptide, 29 amino acids. It's a counter-regulatory hormone which opposes the action of insulin. For postgraduates and undergraduates who may be listening in, just remember glucagon as being the opposite of insulin. Whatever insulin does, glucagon does the opposite. So we know that there are many counter-regulatory hormones which increase the blood glucose level, like epinephrine, norepinephrine, cortisol, growth hormone, and glucagon. But there's only one which reduces the glucose, and that's insulin. And the other thing that you should know is that you can actually have a measurement of glucagon. It's not necessary to do it in clinical practice, but if you measured it, the basal level in the fasting is 100 to 150 picograms per mil. It has a half-life of about 6 minutes. And the secretion rate and so on are shown here. Now, there are a number of activators of glucagon, and the kind of things which activate glucagon is a low blood glucose level, because a high blood glucose level activates insulin, then the low blood glucose level activates glucagon. The other stress hormones activate glucagon. Amino acids can also activate glucagon. Various GI hormones, cortisol, exercise can activate glucagon. Infections can increase glucagon level. It's one of the reasons why you develop ketosis or ketoacidosis after infections. There are many inhibitors of glucagon, of which glucose itself, when the glucose level goes up, the glucagon kind of shuts off. Somatostatin is a natural inhibitor of glucagon, and we'll see that in a minute. Insulin, of course, inhibits glucagon as well. And that is summarized well in this beautiful diagram, which is there in Lefebvre's paper. So there are different kinds of control of glucagon. One can be the endocrine control, which can come through either through insulin or from somatostatin or even the pancreatic polypeptides, which means that all the other hormones secreted by the pancreas actually inhibit the alpha cell glucagon. But you also have the autocrine regulation, which comes through the GLP, and that's what is used in as pharmacology today to treat. There's also other endocrine regulation. Nutrients can affect it. The autonomic nervous system activates glucagon. And of course, you have paracrine regulation as well, which means that within the cells itself, the alpha cells and the beta cells and so on, you have a regulation. So it's a pretty complex regulation by various mechanisms: neural, endocrine, paracrine, and so on. The actions of insulin are several, and as I said, very simple way to remember, it's exactly opposite to insulin. It's a hormone of energy release, and we'll talk about that. Mobilization of stored nutrients, and one of the things it releases is ketoacids, and that's why you need glucagon basically for ketoacidosis, and I'll show you the application of that in a minute. But let me talk about very briefly in 2 or 3 slides about its action on carbohydrate metabolism, protein metabolism, fat metabolism, and others. Now, in relation to carbohydrate metabolism, glucagon increases the glucogenolysis in the liver, but not in the muscle. And these are the various actions of glucagon. So you can see that it works on the liver, it works from the pancreas, formation of glucose from lactate, pyruvate, it needs it, increases gluconeogenesis. All these are the actions on carbohydrate metabolism. As far as fat metabolism is concerned, it increases lipolysis and the fat breakdown. And that's the reason why ketogenesis also occurs. It also increases the release of free fatty acids and glycerol and prevents the synthesis of triglycerides. So glucagon is truly a ketogenic, and that's where the relation of intermittent fasting and so on, where glucagon goes up and then ketogenesis occurs. And that's the basis of several of the treatments that we do. Protein metabolism, it has a calorogenic effect, and it lowers the plasma amino acids, increases glucogenesis by transport of the amino acids. So you can see, like insulin, it has its action not just in carbohydrate metabolism, but all the other metabolism. Not only that, it has many other actions. It inhibits the renal tubular sodium reabsorption, it results in natriuresis, and it stimulates the secretion of growth hormone, insulin, and somatostatin, because these hormones are all linked to each other. It's related to appetite as well. Now, this is a very simple way to remember this, that when you have insulin, you have all these actions, increased glucose oxidation and glycogen synthesis and so on. And these are the actions of insulin. And you can see that in the fed state, insulin dominates, whereas in the fasted states, glucagon comes. It's natural, isn't it? When your glucose levels start going up, you get more of insulin. When you eat, when you're fasted, you don't get much insulin to prevent hypoglycemia, and glucagon then comes up. It's a body's way of trying to prevent hypo and hyperglycemia, which is why in a normal person, the glucose levels remain in the tight, narrow range. Now, glucagon as a hormone of energy is very important because it strongly activates the liver's ketogenic machinery. So whenever you have glucagon associated with ketogenesis, And this ketogenesis is very important when you consider its role in diabetes. And I'm going to talk a little bit about fibrocalculous pancreatic diabetes or secondary diabetes. I'll just touch upon type 1 and type 2 diabetes. And of course, I'm not going to discuss glucagonoma, which is a tumor which comes from the glucagon cells, which can be a deadly kind of a tumor, very often fatal as well. So let me talk about its role in secondary diabetes and fibrocalculous pancreatic diabetes. Now, when I was a postgraduate student, it was believed that all forms of secondary diabetes are usually protected from ketosis, and particularly the fibrocalcific pancreatic diabetes. And this is a figure which I actually made almost 30 years ago to show the heterogeneity in fibrocalculous pancreatic diabetes. Now, the majority of patients with FCPD, which is secondary form of diabetes due to pancreatic stones, are ketosis resistant. They require insulin, but they're ketosis resistant. And that's how they differ from type 1 diabetes, where you get ketosis as well. Very small number of patients respond to diet and oral drugs, very small, and eventually they'll need insulin. Of course, a very minute percentage can even be ketosis prone. But the characteristic or the hallmark of FCPD is ketosis resistance. For years, we did not know why this was so, but we know that they have blood sugars of 500, 600, but they don't develop ketosis. This is my first international publication. I'm very proud of this paper because in 1983, just after my MD, I published this. I just raised the thing, could it be related to the C-peptide, that they just have enough insulin to protect them from ketosis? So I measured the C-peptide levels, stimulated C-peptide levels in non-diabetic people, type 2s in FCPD and type 1. You can see in the type 1s, where they have virtually absent C-peptide, they're prone to ketosis. Type 2s who have better C-peptide, they don't develop ketosis, but they respond to oral drugs. And of course, the normal people have more. But FCPDs were bang between the two. So we hypothesized that they have just enough insulin to prevent ketosis, but not enough insulin to respond to oral agents. And this has, of course, been confirmed by many people. But then we went on and said, what about glucagon? Can glucagon be playing a role? And this is a paper actually published in DRCP in 1990. So we know that in normal people, if you give glucose, what happens is that the glucagon gets suppressed. And we know that in type 1 diabetes and type 2 diabetes, if you give them glucose, the glucagon level will go up. And that's what we found here. This is the glucose level GTTs, which we did in our labs. And we measured the insulin as well. This is normal people. And of course, insulin is reduced in both FCPD and that time type 2 was called as NIDDM. But look at the glucagon level. Now, if you look at the glucagon levels in normal people, it actually goes down. It gets suppressed when you give them glucose. But in type 2 diabetes, it goes up. And in type 1, it goes up even more, which is why they develop ketosis. So our hypothesis was that maybe the alpha cells are also affected and they don't have enough glucagon. And we were right, because you can see a flat curve there. They don't have glucagon. So even if you give them glucose, they don't respond at all. Just like the beta cells are damaged, the alpha cells are also damaged due to the stone sitting there and due to the fibrosis, and therefore they don't develop ketosis. So this is one of the proof that we had that glucagon is actually responsible for ketogenesis. So we concluded in a later textbook, in the Pickups textbook of diabetes, that the reason why they don't develop ketosis is partly because of the partial preserved beta cells, because there is also glucagon deficiency, but also we came up with other theories like carnitine deficiency and low adipose tissue mass. They don't have enough fat, and therefore they're not able to produce ketosis. And it's these four things which produce ketosis resistance. We did one more study where we said, okay, if normal people, the glucagon level gets suppressed when you give them glucose, what happens in the pre-diabetic situation? And there was no study at that time. We're looking at children of diabetic parents who had a normal OGTT. And it's very interesting to see that if you did not have a family history of diabetes, if you gave them glucose, the glucagon levels went down. But among the offspring of diabetic, even before they developed diabetes, you can see that there is no reduction in the glucagon. So already there's a defect in the alpha cell, which we are able to show years before they actually developed diabetes. Now, this is a kind of a cartoon to sum it all up, that in type 1 diabetes, the role of glucagon is that in the treatment of insulin-induced hypoglycemia, even today for children, mothers carry the injection, we can just inject. If they're not able to, they're in coma and they're not able to give them oral glucose or even IV glucose, just give them this glucagon shot. I'm not even mentioning that glucagon is actually used as a test for C-peptide, the 6-minute glucagon test. You do a fasting fasting C-peptide, give glucagon, and after 6 minutes, you can take another sample to see the stimulated one. Contribution to ketoacidosis is well known, and I talked about that. Now, as far as type 2 diabetes is concerned, this is going to be the whole discussion, because now we know that the inhibition of the secretion with the GLP-1 receptor agonist not only helps to control diabetes, but also to reduce weight, as Dr. Shashank Joshi mentioned. And its contribution to both fasting and postprandial hyperglycemia, I already mentioned. And then, of course, when you have starvation and so on, glucagon is stimulated. So to summarize, the uses of glucagon, ladies and gentlemen, is to, number one, manage hypoglycemia, inhibition of glucagon secretion, for which we have the GLP-1 receptor analogs, and we have so many classes of them now, from liraglutide to dulaglutide to semaglutide and so on, all of which will be discussed. We have the glucagon receptor agonist. We now have the dual GIP-GLP-1 receptor agonist, the tirzepatide, and then we also have the triagonist or the retatrutide coming in now, each one getting better than the other. And the kind of weight reduction that you're seeing with the dual and the triple is unbelievable, losing 15-20% of body weight, just like you do bariatric surgery. And I'd just like to end with those of you who want to get in touch with me, those are my contact details. So I'll stop there, and if there are any questions, I'll be happy to take them. So over to the chairpersons. Thank you very much. Thank you, Peter.
Thank you, Mohan, for a very high enlightening lecture, very knowledgeable as always. If there are any questions, because we are already running short of time, perhaps one or two burning questions.
Anil has already raised his hand.
Yeah.
Can I ask you a question, Mohan?
Yes, Anil. Yes, Anil.
We have a lot of patients who come to our clinics and who go to various clinics who give keto diets for weight reduction. And one of my very senior colleague, Dr. Janak Nathan, who was given an award in USA for prescribing keto diets to young children who had epilepsy. As a treatment for epilepsy, the keto diets were prescribed. What is your opinion of that?
Oh, that's a very well established. In fact, that's one of the indications for, of course, we've got much better epilepsy drugs now. But in those days, producing ketogenesis was one of the treatment for epilepsy because of the brain connection. And so even today, keto diets are given for that. And of course, needless to say, for weight reduction, keto diets are being given. In the Indian context or the South Asian context, with the very high carb diets that we have, my only reservation about these diets is that, number one, they are unbalanced because they end up with a lot of fat. And secondly, they're very useful in the short term, but in the long term, the sustainability is a bit questionable. And third, because of the very high fat diet, then they may increase the risk of coronary artery disease and the lipid levels going up. But for the treatment of epilepsy, there's no doubt at all that it was one of the standard treatments, the ketogenic diet was used.
Thank you. Thank you.
Thank you, Asha, for that nice comment. Thank you. I think in the interest of time, if there are no further questions, we'll listen to Shashank. Over to chairpersons.
Banshi, are there any questions from attendees? Because we cannot see the participants.
No.
Right. So shall we move to the next talk? And next speaker is also doesn't need any introduction. His CV is full of publications, more than 1,000 publications. We are very good friends in DASG and IDF. Shashank Joshi, over to you, please.
Thank you so much. We are switching gears from glucagon to circadian biology. And we are switching to biological clocks. Okay. So, after this very elegant talk of Dr. V. Mohan, I am switching gears and talking on circadian rhythm and metabolism. We know that, biological clock and circadian rhythm and disruption in circadian rhythm is responsible for cardiometabolic diseases, and diabetes is no different. And when we look at our biological clock, we are now at, in India, it's around 8:39, when the melatonin secretion is going to start kicking in. And then the bowel movement will be stopped. We'll have midnight, we'll have deep sleep at 2 o'clock. And then in the morning, there'll be a sharp rise of blood pressure after a nocturnal dip of blood pressure. And then we'll have again the stoppage of melatonin secretion. And that is how our biological clock is ingrained. So, whatever we see during bright light, which is sunlight, is going into the retina, and it's getting processed in the seat of our circadian rhythm, which is a suprachiasmatic nucleus. And the suprachiasmatic nucleus is controlling fundamentally the temperature, the endocrine orchestra, the autonomic nervous system, sympathetic and parasympathetic. And that is actually controlling our ability to become sedentary, or is controlling our appetite and food. So fundamentally, we may think that this is very innocuous, but we are all having bright light. Right now also, I'm in my clinic and there is bright light. And fundamentally, the light and dark cycle of the day is the circadian cycle. And every cell of our body is a molecular clock. And that determines the cell proliferation, energy metabolism, and apoptosis. So if you look at the sleep cycle and its relationship with energy, this is an old paper in JCI. If you look at the earlier, plants, fungi, cyanobacteria, the energy availability is based on light and dark. There is a lot of energy during daytime, and when it is dark, it is not available. Similarly, in the metazoa, there is a cyclical energy harvesting. So when you are awake, you're feeding, and you're not feeding when you're not awake. So as I told you, this is how our circadian rhythms are ingrained. And these circadian rhythms have periodicity. And they are across the whole animal kingdom. And this periodicity is circannular, which is 29.5 days, or circumsemilunar, which is 14.75 days. And then it is stimulus controlled or clock controlled. For example, we have high tide and low tide. We have night, darkness, and light. And then we have biological rhythm. We have oscillations. And that is the whole clock orchestra, as it is called. So in an ant lion, there is a circannular rhythm. And that is linked to full moon and new moon. And we all know sun and moon, even religiously we know, has a lot of significance. And then we have the circannular rhythm, which is now we are wanting to fall asleep. But some parts of the world, like where Peter is, it is midday. So, it is the different thing. Then we have circantidal volumes. And then we have rhythms which are circannual in the animal kingdom. And then we have the migration of birds, which is from tropics to colder climates. For example, I live in the city of Mumbai. And at this time, from Siberia, a lot of birds come to Mumbai. And they travel almost 2 months to come to my place, some of these. Then we have ultradian rhythms. And therefore, every rhythm of our body has a genomic basis. It is controlled by a pacemaker or a biological clock. It is calibrated by the dusk and dawn phenomenon, which are photic signals, or activity and rest signals, or the nociceptions or non-photic signals like silence. And then there are these synchronizers, which are the environmental factors. These synchronizers are also German. People describe them as Zeitgebeugers or entraining agents. And therefore, the range of the biological clock rhythm by the Zeitgebeuger will vary, and it can actually be measured. And therefore, the biological rhythms will have properties from unicellular eukaryotes to human beings, which are very complex. And therefore, fundamentally, how is biological rhythm controlled? The Zeitgebeuger is the master controller. There are sensory perceptors, and the clock will affect our locomotor pattern or activity pattern, feeding behavior, hormonal release, including gut peptides and others. And the rhythms are of different times. Either they are trapped with the Zeitgebeuger of 24-hour, which is circadian, or they are free running, or circantidal, or circannular, or so on and so forth. And they control the body temperature, the urinary volume, the cerebral blood flow, the blood pressure, the melatonin, the cortisol, the TSH, and the growth hormone. And this rhythmicity is maintained for various activities of biological functions. And it is the pineal gland, which is the location of the biological clock. In birds and reptiles, it is directly stimulated. In the frogs, it is having a well-developed parietal eye. In the mammals, as we evolve, it gets ingrained in the suprachiasmatic nucleus of the hypothalamus. So the central clock, which controls our biological rhythm, is a suprachiasmatic neurons. And there are a lot of projections of these. These control the preoptic area, the tubulomammillary hypothalamus, the thalamic nucleus, but mainly they control the sympathetic and the parasympathetic response. And from a metabolic standpoint, diabetic standpoint, they control the white and brown adipose tissue, the thyroid gland hormone, the pancreatic beta cell secretions. And that is how the clock works. This is how the clock loop comes. And these are the endocrine axes. So we have external output of light, food, social interaction, and drugs. And then we have behavioral output, which is locomotion, activity, sleep, and food activity. And there are cues which retrain the clock. For example, round-the-clock care by nurse bees is trained by various methods. And we can reset the clock. For example, food or feeding regimens, they will have a role not only on the suprachiasmatic nucleus, but also on the peripheral clocks like the adipose tissue, the liver, the muscle, and various sleep-wake cycle and activities are there. So between sunset to sunrise, there is a melatonin onset, midpoint, and offset. And that really is relevant for maintaining our clock and maintaining our hypothalamo-pituitary adrenal axis. And this determines our glucocorticoid rhythm every day for the various activities of our body. And this is linked to stress. So when you are under stress, for example, Bansi is under stress right now because he has to make me finish on time because there are a lot of panel discussion which you are going to listen. Then there is disruption of the diurnal oscillation of the CRH AVP activity and the ACTH activity. And once that is disrupted, the clock gets reset and the programming of the clock gets disrupted. Normally, the glucocorticoid clock is working in tandem and also is interdependent and is having a very good apparatus for clock-to-clock interactions. And these are transcription processes. And these are sites of proteins like acylation processes. Similarly, in the gonads, we have clock control on gonadal rhythms, whether it is the testes or the ovaries. And therefore, the reproductive outcomes, whether it is ovulation, pregnancy, are linked. And if you are a shift worker, and we have all become shift workers now, due to the disruption of our clocks, we will get disruption of reproductive outcomes and negative reproductive outcomes and pregnancy outcomes. Similarly, it affects the sleep because what we eat and what we spend, because ultimately obesity is an equation of energy intake and energy expenditure. And that is directly controlled by our suprachiasmatic nucleus and the peripheral clocks of our body. And these connections impact metabolism through autonomic pathways, through gut incretins, through metabolism, and through various other gut peptides. And this whole orchestra is extremely well studied. And the core metabolic functions, whether it is inflammation and meta-inflammation, adipocyte differentiation, gluconeogenesis, all of them are regulated by specific areas like CRY1, PER2, which are there, which are responsible for the biological clock, which then have a role on glucocorticoid receptor, PPAR gamma receptor, HNF4 alpha receptor, or the cortisol receptors. And there, if they are disrupted, then you will have metabolic abnormalities. And ultimately, it will affect our food behavior. You may feel hungry or you may feel full. That will be there. And what happens when there is disruption is the peripheral central clock apparatus is disrupted. We start eating too much, we get craving for food. And when we get craving for food, we accumulate fat in the liver, in the skeletal muscle, in the pancreas. So we get fatty pancreas, fatty liver, intramyocellular fat, and we get excess adipose tissue. And that fundamentally disrupts and causes diabetes. So various disruptors when they disrupt our biological curve, you get glucose homeostasis and challenges. And these metabolic hormones get disrupted. We get various clock control disruptions, and they are impacted by sleep. And when feeding timing changes, that loss of rhythmicity leads to diabetes. So that's very, very elegant work. This was Polanski's very elegant paper in New England Journal of Medicine, which shows that when we lose the rhythmicity in diabetes, it is very, very well documented. So fundamentally, the glucose intolerance which we get is due to circadian misalignment. And Frank published this paper, and this is all linked to disruption of the central clock in response to insulin. And the disrupted clock gene leads to gluconeogenesis. It makes us eat a lot of carbs and fats. And fundamentally, this is what happens. We get into this cycle of hypothalamic inflammation. There is hypothalamic inflammation, there is insulin leptin resistance, there is weight gain, we eat more. Similarly, even in the periphery, there is cellular inflammation, insulin resistance, glucose intolerance. So we have the activation at the hypothalamic level and at the peripheral level due to disrupted circadian rhythm. And this is the key. And these circadian changes in insulin and glucagon are documented in the field of diabetes almost a decade back. And it is even the timing of food. For example, the breakfast, the lunch, the dinner, you can see the role of glucose, insulin, and glucagon. And these are the circadian changes. You can see insulin, glucagon, and the glucose. They follow different pathways. Glucose and insulin is in the morning, and glucagon is more in the afternoon. And the insulin-glucose rhythm is intact when the suprachiasmatic nucleus is intact in rats. But when it is disrupted, you can see the curves have changed. And therefore, the clock gene of glucose and insulin is very well documented. And even in the islet cells, there are clock genes. And they also have been studied. And the alpha cells regulate insulin secretion. That also has been studied very well. And that's something which we document very well. And so is the beta cell death. So we have a islet circadian clock also. And this is a paper on the islet circadian clock with the PER1 or the insulin and so on and so forth. And this clock can also get disrupted, and you can get various disruptions and disruption of pancreatic function and beta cell apoptosis. The main topic which I'm going to talk in the next 10 minutes is why is sleep necessary? Because if we sleep well, our brain gets tissue perfusion. If you're asleep, 60% increase occurs in the interstitial space. You get better CSF flow, and all the negative metabolites in our brain get cleared. If we sleep less, particularly less than 4 or 5 hours, then the interstitial space on the cortex is reduced. You get restricted CSF flow and metabolites accumulate. And that causes a lot of problems, including dementia, which comes up, and so on and so forth. So Underwood published this very nice paper. I would urge each one of you to read it. I will send you the full text, which says sleep is the housekeeper of the brain. And there's a triad of sleep, circadian rhythm, and metabolism. And that misalignment of sleep and circadian rhythm leads to insulin resistance, meta-inflammation, and beta cell dysfunction. And when we restrict sleep, the human transcriptome is restricted. And this is data, published data, to suggest that almost a decade back. So when there is sleep alteration, the insulin sensitivity is blunted and you get insulin resistance. So this is the global data published of lack of sleep and risk of diabetes. So less you sleep, more is the risk of type 2 diabetes. And therefore, you can see even post-COVID, we have seen a spurt of diabetes coming from. Short duration of sleep has an odds ratio of development of type 2 diabetes with a factorial of relative risk of 1.28. And therefore, it is important. And even if you sleep too much, you will get sleep. So you need to sleep at least between 5 to 10 hours, and 7 hours is optimal time of sleep. Quality of sleep also matters. And this is data to suggest that. So it is important that sleep disruption is relevant, and it is important to recognize that incidence of diabetes is linked to sleep and sleep disturbances. If you get fragmented sleep, disrupted sleep, that is also related to type 2 diabetes, and there's data to that. When there's rotating night shift, obesity goes up, and this will lead to risk of diabetes and circadian myelin. The other thing is we have chronotypes now. Are you a lark? Are you an owl? Okay, are you a night sleeper or are you a day sleeper? And what are you? Okay, the eveningness, if you are a late light patif, it is associated with increased risk of metabolic syndrome or diabetes with the odds ratio of almost 2. So remember, that is something which we need to recognize. So chronotype is directly linked to glycemic control also in people with diabetes, as well as there's a link between obstructive sleep apnea and diabetes, which is very well recognized. And disordered sleep breathing is also linked to gestational diabetes. So clock is linked, and the main biological activity is acetylation. Sleep has peptides and melatonin. The chromatic remodelers are in the epigenetic control. The cell cycles have the P53 and F-box proteins. Metabolism has nuclear receptors and coactivators. And the clock machinery has BMAL. So this is a very simplistic model of all the basic science work which is happening in clock biology. And due to disruption of lifestyle, the signaling and metabolism, the clock chromatin complex falters, and there is circadian disruption. So circadian rhythm is very important. And we know that heart rate, blood pressure, and cardiac output increases during day and falls during night time. There is a nocturnal dip of blood pressure. We know that there is absence of nocturnal dipping is called as non-dipping. It is linked to target organ damage and increased cardiovascular risk. Early morning blood pressure surge is linked to myocardial infarction, stroke, and sudden cardiac death and ischemic episodes. And the blood pressure variability is also important. So fundamentally, we need to synchronize our food, our physical activity, our social professional interactions, our central nervous system, our steroid and melatonins, our molecular type, our light exposure, including sunlight exposure, and rest, activity, and routine. And if we can align our clock with behavioral changes or pharmacological changes, we can have a better quality of life. This was recognized by DASG by putting some simple things, the 5 S's: sitting, stepping, sweating, sleeping, and strengthening. Just sitting is the next smoking, and break your sit. Sweat it out. Sweat it out. Build some muscle mass if you are frail. At least 500 steps will decrease cardiovascular mortality by 2 to 9%. And 5 to 6 minutes of brisk walk is equal to 4 years of a larger lifespan. Strengthen your muscles. Even if you do yoga or Tai Chi, it will improve insulin sensitivity. But the main thing which came out in this guidance of ADASD is sleep not more than 8 hours and not less than 6 hours, because it negatively impacts the A1C. Quality of sleep matters. If you have sleep apnea, insomnia, or restless leg, correct it. It is an adverse impact on type 2 diabetes. And the chronotype matters. If you're an evening chronotype, you're a night owl, you go to bed late and get up late, you are more susceptible to poorer glycemic outcomes and physical inactivity than a morning chronotype, which means if you're early bird, go to bed early and get up early. So you need to assess sleep. We can assess it by questionnaire or apps. Professor Peter Schwartz is a big world authority on digital medicine and apps. We can track the clock. We can trap, but melatonin is our clock regulator. And it is very important to recognize melatonin very well. Melatonin is nothing but a tryptophan derivative. And it is moved into the process through the whole serotonin pathway. Melatonin is a very easy source. You don't need to take a melatonin gummy. You know, you have a cherry or a dark chocolate above 70%, it has melatonin. Okay. If you have a banana or a ginger or any of the cereals, you will have sufficient melatonin for the day. Peanut butter, walnuts, almond, pistachios will have melatonin in it. If you have a green tea, spice, herbs, you will get melatonin. Okay. The other important thing is at night when you go to sleep, the light emitted by the television or the telephone will shut off your pineal gland. And the best light for bedroom is the red light, because in red light, maximum melatonin can be secreted. So the bedroom light should not be a blue light or a green light. It should always be the red light from a physiological, circadian biological standpoint. The other thing is we should avoid tea, coffee, or colas because they switch off the melatonin. And we should have an early dinner, which is there in many cultures. The other things is we should adopt a sleep hygiene. At least 6 hours of sleep, maintain a sleep wake time, wear loose, comfortable clothing, take a bath, listen to music, shut off the various other things, take your blood pressure and sugar medicines on time. Don't enforce sleep. Avoid coffee, avoid alcohol and nicotine. Don't have junk food or heavy dinner. Avoid all this digital detox you should do. Half an hour before going to sleep and half an hour after getting up from sleep. Don't see your WhatsApps. Don't see your mobile phones, your computer screens, or your TV screens. They are not good for health. They disrupt your biological clock. The light from street lamps, clocks, mobile phones, DVD players keep you awake. There are a lot of superfoods. I don't want to go into superfoods which can make you fine, and avoid all these bad foods. Music is very therapeutic. Music is very therapeutic. So fundamentally, if we want to live long and correct metabolic health, we need some degree of melatonin and we need adequate sleep. So in my mantra for metabolic prevention, apart from eating slowly, eating less, eating on time, the biology of eating is very important. If you want to lose weight, have a large breakfast, moderate lunch, and a light dinner or skip it. There's a lot of biological sense to it. There's a lot of biological basis to it. And there's a circadian ingredient. So it is important to eat slowly, not fast. It is so that you don't get an insulin spurge. You need to eat on time. You need to eat less. But you need to have a large breakfast, moderate lunch, or skip it. We need to move more and do some yoga. But we need to sleep well and sleep on time. And that's very, very important. We must do 1 hour of digital detox every day. And we are all here so that we can have health and happiness. And that's our commitment. So I have tried to give you a very fast glimpse because Bansi gave me only 20 minutes. He says, Shashank, you have to finish everything in 20 minutes. And to finish anything in 20 minutes is not easy. So I tried to give you some glimpse on biology of circadian rhythms, how food and sleep can impact it, how we need to change the way we think. And it's important to recognize that we need to sleep well. So my only take-home message to all of you is that 1, to correct your clock, sleep for at least 7 hours and sleep on time. Second thing is be happy. And the third thing to correct your clock is half an hour before going to sleep and after getting up from sleep, do a digital detox. Don't see your mobile phone, your television screen, your internet gaming screen, or any form of a digital device. And you will be much more happier. We are, as I told in the beginning also, that I'm grateful to Dr. Sabu and the DASG and IDF Southeast Asia, Dr. Azad Khan and Dr. Abdul Basit and everybody here, that we are here for people living with diabetes. But we are digitally intoxicated. We are living in all artificial light. We have disrupted all our biological clocks. And we have led to a pandemic of diabetes. And we can all correct it by doing simple lifestyle measures in which biological clock or circadian disruption has a huge role to play. And we can generate melatonin not pharmaceutically, but physiologically by appropriate foods and by simple lifestyle tips and some good sleep hygiene habits and probably by some physical.
Again, for summarizing, as he said, in 20-25 minutes, the topic which ought to be discussed for an hour or so. In the interest of time, I now request Professor Azad for his comments and to start the panel discussion then.
Sir, you have to unmute. Dr. Anil Boraskar, you can ask the question or comment. And Dr. Azad Khan, you have to unmute yourself. Yes, Boraskar.
Shashank, I just want to know, many patients during the COVID time, I had put them on meloside, melatonin. And they had a good sleep. And in fact, many authorities, including you, had suggested that meloside or melatonin tablets, 3 milligrams given at night, does not cause any hangover feeling. It gives a good kind of sleep. The quality of sleep is better. And it also protects in the pro-inflammatory cytokines due to COVID. I mean, this was a paper I saw, and I personally treated many patients of COVID with melatonin tablets, and they did very well. What is your experience on that?
No, so I completely agree. See, what happened during COVID, people were under stress. So they were not able to get sleep. They became restless. So by giving melatonin to them, they were able to sleep better. And if you just sleep for a good 7, 8 hours, just right, okay, automatically it is anti-cytokine. It is therapeutic. Sleep is therapeutic. So, but sleeping adequately is important. Sleeping too much is also counterproductive. So sleeping right is important. And all melatonin does pharmaceutically or physiologically is resets the biological clock. But it's a stopgap measure. We use it for shift workers, but a lot of science revolves around
There is
getting the
also a
sleep
recommendation,
hygiene.
Shashank,
Yes.
that if you want to, you have a very busy day tomorrow, then it is better that you sleep alone. And ideally, the spouse should sleep in other bedroom. Is it really true?
I don't have any scientific answer on that, but
No,
I
but
am not so sure.
undisturbed sleep in the bed where there is no movement of your spouse, does it improve the quality of sleep?
There is no evidence-based answer to this. We can have a lot of jokes on it, and we can digress, but we have a very elegant panel discussion. But scientifically, I don't think that's
Yeah, thank you.
I think it is important that we need bedroom hygiene and bedroom discipline. But I think if there is intimacy, it is also therapeutic. So I may not agree with what you're saying, and there is data, but it is beyond scope of this talk to suggest that intimacy in bed also keeps people happy. See, ultimately, if you get adequate sleep and you're happy, I think it is reasonable. But if there is stress generated and there is a non-intimate hostile environment, then probably what you're suggesting and recommending may be appropriate. So I think it is situational and it is individualized. Professor Azad Khan, sir.
Yeah, I said that I like your sentence, digital detox, yoga and yoga. That will do the trick. Thank you.
Perfect. Thank you. Thank you, Shashank. Thank you, Dr. Vimohan and Professor Azad Khan and Dr. Abdul Bashir. As we have thought that, one talk we will keep dedicated to glucagon and one will keep related to that. And that's what we asked Dr. Shashank to talk on circadian rhythm and how it can lead to metabolic issue. With this, now we start with us a panel discussion. The moderator is Professor Dr. Brij Bakkar, who is currently president of RSSJ, as I already introduced him, and he is director of Dr. Bakkar Diabetes and Obesity Center from New Delhi. And he have eminent three persons from Peter from Germany, Sharad from Qatar, and Dina from Nepal. So he will introduce all three and then he will start the panel discussion. We have 20-25 minutes for panel discussion. In between, if any question come, I will directly message Dr. Brij Bakkar on his WhatsApp. Over to Dr. Bakkar.
Thank you, Dr. Bansi. And once again, I welcome you to this panel discussion, Targeting Obesity, the future of prevention of all non-communicable disease. And for this, we have three very eminent panelists. We have with us Dr. Dina Shrestha from Nepal, who is Senior Consultant Endocrinologist at Norwich International Hospital and is also the President of SAFES, which is Southeast Asian Federation of Endocrinology Societies. And we have Dr. Professor Peter Schwartz from Germany. Can we have the slides? Okay, we have Professor Shahrat Tahri, who is a senior physician from Qatar, and we have Professor Peter Schwartz, again a good friend from Dresden, Germany. He's president-elect of IDF also. So I think we are running behind schedule, so I'll not take much time to go through the detailed introductions, and let us move straight to the topic of targeting obesity to prevent maybe all the non-communicable diseases in future. And moving on from the previous presentations, one on glucagon and one on circadian rhythm, I would go to Peter, and Peter has a lot of experience on lifestyle interventions and digital, markers for non-communicable diseases. So, Peter, what is your take on focusing on these two important parameters? One is the sleep or the circadian disruptions that we are having because of the modern lifestyle, and the upcoming role of targeting glucagon receptor in management of obesity.
Yeah, thank you very much for this question. And thank you very much for this excellent meeting, this excellent presentation, because this is what we have to do, share our ideas, share new knowledge, and think how to apply this knowledge to our daily clinical care. And this is basically already the bridge to your question. In my eyes, glucagon, like Dr. Mona has said, it was a stepson treatment. And this was exactly our failure in the past. There was insulin. We were focused on insulin, treating at the end only glucose. But we forgot that glucagon is the same as important and maybe even more important in the pathogenesis of diabetes compared to insulin. Diabetes does not only develop due to the lack of insulin or insulin resistance, it also develops due to the disruption of the glucagon metabolism. And it was on one slide of Dr. Mohan, it's the paracrinopathy which is important. So the interplay between the alpha cell, the beta cell, or let's say the alpha cell and the beta cell. And there are new data that the lack of intracellular insulin, that means the amount of insulin in the beta cell, is directly related to the release of glucagon. And now we could say, okay, it's clear, we know this, but if you have someone with type 2 diabetes with a severe insulin resistance, the beta cell is producing insulin, and it's immediately released to the cell membrane and released out of the cell. So the intracellular insulin goes down, even if the patient is severely insulin resistant, and this pushes the release of glucagon. So we have high insulin and high glucagon. And this is the devil if you talk about obesity and diabetes. And why I'm saying this so long, and coming back to your question, in my eyes, this is the target. If we think about future effective therapies, this is the target if you talk about prevention of diabetes, prevention of obesity, but also treatment of diabetes and obesity. And our laziness in understanding the glucagon, I think, should be directly over with the discovery of GLP-1, GIP, all these good new drugs. Because why are they so good in helping the patient to lose weight? Because they target the glucagon metabolism. And now we could say, okay, we have good drugs and everything is solved and diabetes is cured. It's not like this, because our obligation, our ethical obligation, is to apply this knowledge for prevention of diabetes. And this is a link in my eyes from glucagon to the presentation from Shostak, because the glucagon metabolism and the circadian rhythm are tightly interconnected on an endocrine level. And what Shostak just has recommended, sleeping 7 hours, detoxing our daily behavior, is the first step for ourselves to apply the knowledge from the glucagon metabolism and circadian rhythm to the prevention of chronic diseases. And diabetes is not the frontier in this. It's chronic diseases, it's hypertension, metabolic syndrome, the non-communicable diseases. And this, I must say, is a sexy discovery. Please allow me to say it in this, using these words. Interconnecting this endocrine knowledge, applying it to our daily lifestyle and to prevention of diabetes and obesity, this is what we should do, and we should do in an easy way for our patient. And this is my take home from both of the lectures, where I'm very thankful to these two excellent, intuitively practical lectures.
Thank you. I think very important points very important observations that you made. Dr. Tarak Tahari, what is your take on, targeting obesity as the future of prevention of all non-communicable diseases? We know obesity is possibly the mother of all important diseases of adult life. And, I still remember about 20 years back, we used to say about 20 diseases are caused by obesity. And now list is already touching 240 diseases where obesity plays at least a predisposing role, if not a causative role. So, looking at so many diseases where even we don't know the exact mechanisms how they are being caused, what is your take on shifting the focus to prevention of obesity or targeting obesity to prevent all those, fallouts of obesity?
Thank you for that question. I very much enjoyed both talks on glucagon and circadian rhythms. So the first talk should really put things in the picture. So we need to work out how these new drugs actually work in terms of weight reduction. So glucagonoma syndrome, we know it causes rashes and diabetes and weight loss. A lot to do with peripheral effects, but also potentially central effects of glucagon. And then the sleep is obviously, as circadian alignment is very important. So obesity, if you look at when diabetes occurred before, so if you look at type 2 diabetes occurs in individuals who are much older. So if you look at the Look Ahead study, the average age of the people who were enrolled into Look Ahead were about 60 years old. Now, we did a study in Qatar, and you look at the average age, it's about 40. So what obesity is doing is creating accelerated aging. So diabetes is driving people who are already vulnerable to develop type 2 diabetes earlier on. Similarly, you could talk about diseases such as obstructive sleep apnea. Those who have a vulnerable airway, for example, obesity pushes them over the edge in terms of manifesting that disease. So before, what sort of how we treat diabetes now, current view is has been that we give a bit of education to patients, and then we give them drugs, and we give them more drugs, and we give them more drugs, and they're very glucocentric. And if you're very good, then we address their hypertension, and then we address their dyslipidemia. And we're very happy with that because the HbA1c is okay, the cholesterol is okay, and the blood pressure is okay. But the patient's very unhappy because they're getting multiple therapies that are actually making them have multiple side effects. But we don't really address the underlying issue, which is obesity. Because if you address the underlying issue, then we can reduce the patient burden. We don't need to escalate medications. We could reverse diabetes. So we conducted a clinical trial called DIADEM-2 in the UK, where we put people on meal replacements and then introduce food gradually. And actually, these were people with 13 years of diabetes duration on insulin, and 33% of these individuals came off insulin. So it's possible to actually really, and their quality of life improved. Then we asked the next question is, can we go back with people with early diabetes and be able to put them into remission? And we managed to get 61% remission with the same kind of intervention in people with early diabetes by reducing obesity. And particularly, it's been shown through reduction in fat in the liver and pancreas. So it's possible to actually think about type 2 diabetes differently now as maybe a potentially reversible disease, that we should address obesity as a first-line treatment. Now, let's look at the drugs. So most of the drugs that we have now available, they're really exciting drugs, GLP-1s and dual agonists and triple agonists, what we call NUSH now, which is nutrient-sensitive hormone-based therapies. So these have shown great effect in individuals with pre-diabetes to stop them moving into diabetes, and then with individuals with early diabetes to be able to put them into remission. So now we're talking about different targets of type 2 diabetes now. Before we were happy with 7% HbA1c, but now we can actually talk about putting people into remission with drugs. And we've known this through surgery, bariatric surgery, where we've seen clearly that it's possible to have remission in people with bariatric surgery. The question that remains now is how long this is going to last in terms of how long are the drugs going to be sustainable? Are there going to be other mechanisms coming through? But unless we treat the underlying condition, which is obesity, we're not going to succeed. But even, I mean, I do an obesity clinic, and most of the time I can see that eventually there's going to be some failure in the way we treat individuals because of the complexity of the biology of obesity. If you go back and look at these things, it's okay to tell people to sleep 8 hours or X amount of hours, but actually it's all set in childhood. So we know there's a really good relationship between short sleep duration, there's linear in childhood, whereas in adults there's a U-shaped relationship, so longer sleep and shorter sleep in adults associated with obesity. We showed that in 2004. But if you look at children, it's linear. Short sleep duration in children is associated with excess adiposity. And by that time, many epigenetic changes have occurred. So by the time they come to us adults and we're giving them like 100 different things to do to change their lifestyle, it's very difficult to sustain. So my view is that we really need to take an obesity-centric view. And it's, by obesity, I don't mean BMI. I mean by somebody who has obesity as a chronic, persistent, relapsing disease. I'm managing that as a non-communicable disease. That's going to be chronically managed with support and the right medications, right treatments, or surgery at the right time. And unless we change that, we're going to get a whole population who are going to continue to have the burden of diabetes, the lower quality of life, and all the complications of diabetes. So I believe, I'm going to argue here for the other panelists to maybe argue back, is that obesity should be central to how we manage type 2 diabetes.
Thank you for those very valuable comments. And we all know now that if you look at the latest ADA, ESD clinical practice recommendations for diabetes management, obesity has become one of the core, targets for managing diabetes. Now, moving to Dina. Dina, we are in a different part of world when we compare ourselves with Peter or Sharat. In our part of world, obesity is still, considered as a sign of prosperity in some of the populations. And obesity is not seen as a disease at all. Now, knowing from the knowledge that we are getting, I don't think that we still fully understand obesity or diabetes. It's a journey of understanding that we are having for more than 100 years. Maybe it will take another 100 years to fully understand what is happening. But we know currently that if we target obesity, we can prevent diabetes, type 2 diabetes, and we can prevent a lot of other things. And if we intervene in people with obesity, looking at the data from direct trial, we could put people in remission for diabetes. There was no blood pressure medication required. There was better quality of life and so on. So what is your take that, how to overcome this change, this perception of obesity, that it is still considered as a part of affluence and not as a disease. So how to take up this challenge in our region?
And thank you so much. And really, I think that is the most appropriate question for us. I think from in our region, in our culture, South Asia, I think that has been the biggest problem, is that when people have prosperity, when they're good, they think that they need to put on weight, so any person, any parent, any partner, everybody just wants that they should be fed and the weight should go, it should increase. Especially, I think that is why we're facing so much problem with gestational diabetes. And like people say that postpartum we should be able to lose weight. Our women tend to gain more weight, and gestational diabetes people tend to become type 2 diabetes and remain as type 2 diabetic, and that is all because of culture. If you ask me what we can do, is I think for every solution it's education. It's education and awareness, like something that we're doing. Like today, we have to tell people that we are not living in 100 years before or 50 years ago, when malnourishment, diarrhea, infectious disease is what used to kill us. And what people are also scared in our still Nepal is struggling. I don't know about India. I think it's probably also the same, is that we have still a lot of tuberculosis and we have a lot of undiagnosed cancer. So when people start losing weight, when they don't feel good about themselves, they think that they have one of these diseases that are more life threatening. And actually, when they put on weight, they feel that it's good. So even these diabetic patients who come at a catabolic state, they have like 400, 500 sugars, like Dr. Mohan said, and they don't have ketosis, and they lose weight. They primarily come to us to say that they want to gain weight, So, and we are like, no, you've actually, it's good that you've lost weight, because obesity is what causes, or overweight is what caused your diabetes, but it's very hard for them to believe that. And the other thing is also that I think what I see is that in South Asia, our obesity is just very central, and which is the worst kind of visceral fat that we have, which has been proved by so many of your from India. There's so many papers. And so now in my clinic, I don't know if it's appropriate to say it here, but now I actually have a mirror in both my clinics in my hospital, which is only going below the body. So people can look at themselves as the body, that they have visceral fat, not your arms or not your legs and not your face, because that seems to be, you don't look very well. And so they only look at the mirror and they see their face and they feel that they're losing weight, they're emancipated or they're catabolic, but they don't see the central obesity. So my job is, as soon as they come into the clinic, they have to walk to the mirror where they show the lower part of the body, where actually South Asians put on weight. So you're right. Awareness, education, especially adolescent diabetes, is becoming a problem after COVID with all this digitalization, which is a good thing. But definitely, that is the way to go, I think. Thank you so much.
Yeah, I really like Dr. Dina bringing.
Thank you, Dina.
Sorry
Yes,
to interrupt.
sure.
Glad you bring about the mentioning of cancer. So many people are worried about weight loss and cancer. If you actually look at, it's interesting to look at 10-year survival with breast cancer is 80%. 10-year survival with type 2 diabetes is 50%. So type 2 diabetes is a really bad disease to have.
Yes. Actually, obesity, leads to reduction in not only quality of life, but also longevity. And it is more than some of the cancers. Now, another important thing, Dina will agree with me, is that people don't want to refer to another person in family as obese. They will come to the clinic and the wife will say, look, my husband is healthy. So I have to tell them, look, this is not healthy. This is unhealthy. First thing is stop calling them healthy. Start recognizing that this is not a healthy state. And as Dina mentioned, people start losing weight because of tuberculosis or cancer. We still have a lot of undernutrition in our country. There are populations which are still, suffering from undernutrition. So gaining weight is sign of, prosperity or affluence, and that's a big challenge. But I think education is working on that. So coming back to you, Peter, where do you see, management of obesity? Because now we know, as of now, whatever understanding of obesity we have on current day, we are able to achieve weight reductions going in the range of bariatric surgery, that is what 20-25% with the newer molecules. But with the kind of challenges these molecules have, one, they are not easy to produce molecules. Second, the cost involved. And third is the availability. With the kind of number of people living with obesity globally, we are looking at figures going above 1 billion. Where do you see, these molecules, or where do you see that whether this strategy of using newer drugs will be helpful, or we have to focus on more and more education and look for, some more options in the future which are more practical, economical, and usable at global level?
Thank you very much for this easy question where it's not easy to give an answer to it. But I would say you actually have given the answer yourself in the question. One of the challenges is related to prevalence of obesity. Our perception of obesity, this is what Dr. Shrestha has mentioned, has changed over the last years. And this goes along with the increase in prevalence. If we see more and more people who are obese in our family, in our peer environment, we don't perceive obesity anymore as a problem. This is number one, what we have to tackle. But we know from behavior change, if we come with more and more problems, this is not very motivational to change something. So we have to think about how can we address the problem for the people. And the molecules you have mentioned are, I would say, are a dream to achieve a short-term effect. Why I'm saying this? I want to be a little bit provocative. Yeah? These molecules are the dream for the industry, because you give them this molecule, you help the patient to lose weight, but you cannot cease the therapy. So you have to put the patient continuously onto the therapy. There are tries and studies with the washout phase, with ceasing the therapy, switching to metformin alone, or so on. But the patient mostly gain weight again. So is it really the right concept to have an obese person who comes out from a peer environment which is pushing obesity, and then to give a molecule lifelong to reduce body weight? I would say from a preventive aspect, it's not. And again, I would say Dr. Shrestha has given the answer. It's education. But education alone has I would say, a bad stigma that we tell the patient what the patient has to do. In my eyes, it's a motivation to a sustainable behavior change. So the patient or the person with obesity has to identify his own situation as a situation they want to change and develop his own way how to change. And this is a basic behavior change therapy or behavior change concepts. And this is the point where I think that digitalization can be a very, very important cornerstone. Dr. Schauschak, as we met in EASD, he has presented me or told me about a study using digitalization with artificial intelligence and sustainably helping the patient to change behavior and lowering the A1C more than any of our diabetes drugs can achieve. If we learn this more, if we understand this better, if we understand how we can use digital tools to address also different levels of digital literacy of our patient and helping them to change behavior to lower body weight, I think this is the most sustainable strategy we can go. The drugs are excellent to achieve a short-term effect and have also an excellent effect on improving cardiovascular and metabolic outcome. But the last statement, because we started today with glucagon, and in my introduction I said glucagon could be the cornerstone explaining why fasting is so effective. And now I'm talking as president-elect of IDF. We have put together a group based on the very good work from Mohammed Hassanain from the Gulf region about fasting and diabetes. And it's not only Ramadan fasting, it's many different kinds of religious fasting, but there are also many habits of religious fasting will become more and more interesting in the medical field for the patient. And I think that different styles of fasting can become an endocrine backed up, and there glucagon is a very important player, an endocrine backed up strategy to tackle obesity and to enable prevention of obesity and prevention of diabetes at the same moment. And I'm very thankful to the statement from Dr. Shrestha and also Dr. Taheri, especially if we only have to deal with obesity, yeah, we have one good target, but this is not the reality. The patient has diabetes, metabolic syndrome, hypertension, dyslipidemia, different kinds of paracrinopathies, and this makes it difficult. And that's why I believe that using the good molecules is not the only sustainable strategy. We have to focus on behavior change of our patients.
Yeah, so I think for that, I would also like to add that because we are doing that and we are using digital and AI, and we know that children are more addictive to these things. So I think it's a good idea to catch them then and use this
Dr.
against
Sharad,
them. So
your comments on this.
Well, I think behavior change is a good approach, but I think it's really difficult to do. And, you know, many behaviors are set, and it's very, very difficult to make people change their behavior, whether in an environment that promotes this obesity by itself in terms of reduction in physical activity and availability of good foods. It's okay to, I think we've gone circle around this with the idea that it's willpower or behavior or any of these things. There's a lot of biological mechanisms that are involved. I don't think the drugs are the solution to everything. You have to look at the degree of adiposity and what needs to be achieved clinically for that individual, as well as in terms of weight loss. It's all about weight loss. So an individual who's got pre-diabetes, they only need to lose 5% of their weight in terms of preventing the development of diabetes down the line. If somebody has more severe diabetes or more prolonged diabetes, they need to have a bigger weight loss target. So again, it's like managing blood pressure. So if you look at the different degrees of weight and how it's affecting the underlying biology, then we can use these drugs effectively. Now, in Qatar here, we've got 40% obesity, 20% diabetes, but we're happy to give 20% of patients multiple drugs for life, but not to give them enough drugs for their obesity. It just doesn't make sense. So we need to really think about where these drugs are going to sit and how long their effect is going to last. Obviously, these drugs will become cheaper in the long term, and particularly non-peptide agonists as well will be available, which are much easier to produce. But we need to really think about where to place these. But of course, I really agree with everything that's been said, education. But even going before, our colleague from Nepal was amazing to mention gestational diabetes. So gestational diabetes prevalence is going up. So we have 1/3 of pregnancies in Qatar affected by gestational diabetes. I'm sure it's very similar in India and Nepal. And when there's pregnancy, gestational weight gain and gestational diabetes, by the time you're born, the number of your muscle cells are set, the number of your beta cells are set, your brain appetite circuitry is set. So, and they come to my clinic as an adult and I can't solve that problem because they're limited already by the uterine exposure to gestational weight gain and diabetes. So unless we go back earlier and address that aspect, as well as the environment, we're not going to succeed.
The speaker is off. Dr. Bridge? Speaker is off.
Dr. Makkar sir, you have to on your
Yes. So I think we had some wonderful discussion going on, focusing on obesity. But what I think is very important thing that has happened in last decade is that we have a lot of shift of focus on obesity side by side rather than just treating diabetes. And now we are looking at obesity as a target for prevention for multiple non-communicable diseases, thanks to availability of more drugs and more better achievable targets in terms of weight reduction. But I am still not very sure whether we are close to perfection. We are still in the early part of journey, and I am sure it is going to take many more years to be close to perfection in handling obesity and prevention of NCDs. But more important thing is that the journey has begun, and I think we should all continue our thrust on targeting obesity to tackle multiple problems that come out of obesity or that are downstream, complications or comorbidities that are coming from obesity. I think we are already running behind schedule. It's 9:50 p.m. India time, and I was supposed to finish as early as possible. But we had good discussion going on, Thanks to all the panelists for doing such a wonderful job. Let us have a big round of applause for all of them. And over to Dr. Bansi. Dr. Bansi is already back home from his clinic. I can see that. And for final comments.
Thank you, Dr. Makkar. First of all, thanks to all three panelists and Dr. Makkar and both the speakers, Dr. Mohan and Shashank. I can see many of my friends, Asher and many more friends from all over the world who have joined. Just to share a good news that we have 1,139 participants from all over the world, and definitely more than 900 were from India. But I'm happy that 200 participants were from different parts of the Asia, and few of them from Europe and UK and even from USA. Yes, definitely they are very few, but still we could reach out to across the world. That's really, I will say, DSG and IDF Southeast Asia could reach out to many parts of the world. That's really great. And I'm really thanking all the delegates and all the participants, because more than 1,900 actually registered delegations. I'm sure they will be joining for tomorrow. We will have two wonderful talks tomorrow and then one again, a panel discussion. So please don't forget to join at 8 p.m. I'll be again seeing you all who are faculty of today, but I will be requesting all the faculty members if they can join for tomorrow also. I'm once again thanking Diabetes Asia Study Group, particularly President Dr. Abdul Bashid, for giving me all the freedom to do it. I mean, under his guidance, and Professor Azad Khan to give me all the time that, yes, go whatever you want to do. And we are thanking Dr. Mohan also for accepting our special issue, who is getting our special issue in this on 14th of November on World Diabetes Day. And we will be releasing physical copy to all our members who are coming to RSSDA meeting also. So that DSG will be, and GOD will become more popular. Thanking IDF head office, particularly Dr. Peter, who is here, and Dr. Akhtar, for their blessings for inauguration, as well as for joining us as a panel in discussion. And just to share with everyone that Dr. Peter is also a member of DSG and a very senior member of DSG, along with Dr. Azad, along with Dr. Akhtar. So I mean, both the IDF head office is also the member of DSG, and DSG is growing very fast. And to inform everyone, this year, last week of April, we have our DSG annual meeting. Let's say biannually we do meeting at Qatar. So I'll be sharing the first flyer at RSSDA only. We are already in the process of making in some scientific highlights. Me and Dr. Abdul Bashid are working on it, and we will be sharing it in a week time. At the first flyer we will be sharing at RSSDA. With this, thank you everyone from our Diabetes India, RSSDA, and Diabetic Association of India, all three major organizations of our country who are member of International Diabetic Federation. We have with us Dr. Makkar, Dr. Amit Gupta, Joint Secretary, as also executive member of RSSDA, and Dr. Anil Bhoraskar from Diabetic Association of India, and Dr. Shashank Joshi. With these words, I thank once again, and good night, and final words from Dr. Bashid, and then we close the session. Thank you.
Yeah, the final words are after this exhaustive session, which was very interesting. As Shashank has strongly preached, go to bed, have a nice sleep, and it shall be 7 hours of sleep at least, not more than 9 hours, please. And see you all tomorrow. Bye for now.
Thank you.
Bye-bye.
Thank you.
Good
Bye-bye.
night. Good night, everyone.
Good night.
Good night. Good night.